All right, so we're going to move into section 7.6 of Unit 7, which is going to be an introduction to Mendelian genetics. So we're going to see the foundation of genetics as far as tracking traits, and that's going to lead us into Punnett squares, which will lead us into pedigrees. So Gregor Mendel, of course, is considered to be the father of modern genetics. All of his work, or he worked as a breeder of plants and bees, but the work that we're going to focus on and the work that he's most famous for is the work that he was able to do with pea plants, and he paved the way for science to understand this idea of inheritance of traits. Self-fertilization, true breeding. So Mendel worked with pea plants which are normal self-fertilizing or self-fertilizing or also self-pollinating. So self-fertilization means that each plant provides both gametes, both male and female, pollen and egg, the sperm and egg. Mendel used true breeding organisms, which are organisms that always pass down the same trait. For example, tall pea plants that only make tall pea plants are true breeding plants, and short pea plants that only make short pea plants are true breeding plants. Cross-pollination. So in order to mix traits, Mendel crossed true breeding plants of different traits with one another, creating a hybrid. Here's our hybrid, a cat horse. I'll let y'all have fun coming up with names on that. So a hybrid is an organism that is the result of a cross between genetically dissimilar organisms. Cross-pollination is the act of mixing cells of two different plants. So he took true breeding tall plants and crossed them with true breeding short plants. So a trait is a specific characteristic that varies from one individual to another. Traits can have multiple expressions. For example, eye color has multiple expressions in humans. Traits can be tracked using diagrams that show successive generations. P generation is the original parent. F1 is the offspring of P, and F2 is the offspring of F1. So we can track those particular traits through flowers, or really whatever traits you're tracking. We can go from level to level to level. So alleles are traits that are coded for by DNA on genes. Genes have at least two expressions called alleles. Different alleles represent the same gene, but a different physical expression of that gene. Some alleles are dominant and some alleles are recessive, and that's going to lead us into when we talk about with Punnett squares, dominant, homozygous dominant, heterozygous, or homozygous recessive. So Mendel's principle of dominance states this: some alleles are dominant and some are recessive. The dominant allele will always mask the expression, recessive alleles when found together. Dominant alleles are typically represented with a capital letter and recessive with a lowercase letter. You see homozygous dominant capital letters, lowercase letters meaning the recessive trait or recessive allele. Genotypes and phenotypes. Traits can be discussed in two ways: as the genotype and a phenotype. So the phenotype is going to be the actual physical characteristics that you see. For example, color or height or size. Okay, those are physical traits, physical characteristics that you can see with your eye. Genotypes are the genetic makeup, okay, the genetic code. So whether it's homozygous dominant, which is two capital letters, homozygous recessive, or heterozygous, which is one of each of the alleles, a dominant and a recessive allele. Mendel's principle of segregation. So in a cross between tall and short true breeding plants, 100% of the F1 generation were tall, and the allele for the short plant was being masked by the tall allele. Mendel allowed the F1 generation to self-pollinate. In the F2 generation, 25% of the offspring were short. So the recessive allele separates from the dominant allele and is able to be inherited even if it is masked in the physical expression. Right? So this shows us that just because there's dominant alleles present does not mean that the recessive allele will not show itself in some situations. And here we can see that it's a flow chart of that happening. That when the next learning target will learn how to set up Punnett squares, and we'll see that the traits will separate from one another as we go through um, the particular crosses. There is a form that goes with this. If you have questions, let me know. If not, then we will continue on with Punnett squares and then next learning target. Thank you.